So I'm very happy to be invited at Aspire. I'm going to talk about aneuploidy rates, how can they be affected not by maternal age, but also by the ART procedure. First, affiliations and conflicts of interest. I've been in this field for quite some time, so I have plenty of conflicts or potential conflicts. I'm a founder at Overture Life right now, where we are doing automation for IVF. Professor at Yale University, and I'm involved in phosphorus, metansers, homoinvestis, and I'm consulting for copper surgical and ferring. First, I'll review a little bit what's the status of chromosome abnormalities in embryos. As you know, aneuploidy and chromosome abnormalities increase with advancing maternal age, and they are probably the major cause of loss of implantation with advancing maternal age. And this produces the dramatic decrease in implantation rates that we see with advancing maternal age. Interestingly, when we select an embryo that is euploid, that is chromosomally normal, those embryos implant equally well at any age. The problem obviously is that you may not produce any at a certain ages, and therefore that doesn't help. But if we find an embryo that is euploid, they implant equally well at any age, at least up to age 42. We detect chromosome abnormalities now with a bunch of techniques, and the techniques that are being used right now are next-gen sequencing mostly. The latest theme on next-gen sequencing is adding artificial intelligence to do the screening. And as you could see, we can test up to 4 million independent sites. We can analyze up to 4 million independent sites, so that gives us quite a bit of information about small pieces of DNA, if they are present or extra or missing. What we found is that on the same age, basically, there is no effect. Sorry, it's moving. Sorry. We don't see an effect on the number of eggs produced. So within the same group of age, we find exactly the same rate of chromosome abnormalities. However, with advancing maternal age, there is a significant decrease in chromosome abnormalities from 59% in egg donors to 14% in patients over 42. What's important is the number of embryos produced, because the more embryos produced, the higher the chance at any age to have more euploid embryos. So, for instance, with an egg donor, with 4 or 5 embryos, you have enough. But for a woman that it's 40, you need more than 10 eggs to have the security to find at least one euploid embryo. Interestingly, also, what we found is that it's not the amount of embryos that is important, it's also the amount of euploid embryos within that group. And it's not that aneuploidy is increasing. Actually, what we found is that euploidy is decreasing with advancing maternal age. So there is fewer and fewer euploid embryos. Meanwhile, aneuploid embryos seem to be constant. Also, we can detect mosaics with next-gen sequencing, and they don't increase or decrease with advancing maternal age. Although here in this statistic, it goes from 16 to 8%, but this is because some mosaic embryos are classified as euploid because they become aneuploid and mosaic. But mosaicism is not increasing with advancing maternal age, since they occur postmeiotically. There are, in addition of maternal age, there are other causes that could create chromosome abnormalities, and this is the bulk of the talk. to find which elements are producing chromosome abnormalities in ART, so we can improve and produce more euploid embryos. The objective of the study is that we know that in addition of maternal age, some ART procedures could induce chromosome abnormalities, but the problem is that there is lots of variation between clinics in maternal age, in infertility indications. So we thought that a good group to study would be egg donor cycles, because these egg donors are selected in the same way in many countries. All are young, all are fertile, and this way we eliminate a bunch of biases when comparing clinic to clinic. And this will allow us to detect if there are iatrogenic effects on aneuploidy. We did a larger study comparing many centers, and we study egg donor age, infertility reason, day of biopsy, paternal age, other indications, cohort size, and the center itself. And we found that none of these factors affect chromosome abnormalities. Only the center was affecting chromosome abnormalities. Paternal age, for instance, did not affect rates of aneuploidy in egg donors. When, as you could see, from 23 to 25%, irrespective of paternal age. But we did 1,645 cycles of PGT in egg donors from 42 centers. Average age was 25. Average aneuploidy rate was 31. But it ranged from 18% to 60%, a huge difference in aneuploidy rates in egg donors. So some centers produce very few normal embryos, and some centers produce many more. The ones in red were the ones that produce significantly less, and the ones on green significantly more. So what could cause these differences? Well, also in addition of differences in aneuploidy, we have also differences in other genetic abnormalities. For instance, this is a study by Dept, and they found that in addition of differences in aneuploidy, there were also differences in mosaicism. Interestingly, when aneuploidy was low, mosaicism was high in some groups, in others not. Also, there were differences in mitochondrial DNA. Some centers produce more embryos that had elevated mitochondrial DNA, which, according to some groups, this could impair implantation. So what are the causes of these differences between centers? We started by looking at centers that each doctor stimulated differently. There were 7 centers that had several physicians that stimulated the same way. So in some they stimulated the same way, and there were no difference between aneuploidy per doctor. But in others, each doctor stimulated differently. And in those, we saw significant differences in aneuploidy rates per doctor. In another similar study, also we compare aneuploidy rates between physicians, and also there was a significant difference between doctors in aneuploidy rates and complex abnormals. The ones that tended to have more aneuploidy have less mosaicism, and vice versa. Then, because of that, we thought that this could be hormonal stimulation. And there have been 2 studies that have analyzed different parameters of hormonal stimulation. One is McCullough et al. In 2019, and another is Irani et al. In 2020. And they analyze M2 retrieve, estradiol on day of trigger, total gonadotropins, days of stimulation, and the fraction of HMG from the total of gonadotropins. They didn't find an effect on any of these parameters, with the exception of the fraction of HMG in total gonadotropins. This was not analyzed by Irani, so it was only found by McCullough. That doesn't mean that it's not in the study of Irani. And this is very interesting. So it means that euploidy rates were positively associated with the fraction of HMG. In that study, we saw that euploidy increased with increasing HMG, at least up to 0.45. Then it's not so clear there weren't enough dots to calculate it, but at least it increases significantly up to 0.45, which will be the optimal fraction. Another hypothesis is that if it's not hormonal stimulation, it could be the size of the follicles that different doctors are retrieving. So we analyze if the follicle size has an effect on aneuploidy. And what we found is that follicle size has no effect on aneuploidy. It does have, obviously, an effect on maturity. So mature GBs are much more, they have smaller follicular sizes than M1s, and M1s has smaller sizes than M2s, but the size of the follicle had no effect on aneuploidy. Again, follicular size is not related to aneuploidy. Then, just not to blame all the time the doctor, there have been many papers also associating different ART procedures to chromosome abnormalities. One of them is just by simply using one culture media or another. This is one of the first papers showing that depending on the media that you use, you would produce more chromosome abnormalities. This is just comparing Ksome to SAGE. Then variables such as temperature, I found many years ago in 1997 that increase mosaicism with lack of temperature control. Then there's another paper on the same theme, Hogan et al. In 2014. They did not find a difference, but they were comparing only 36 versus 37 degrees. We were comparing 25 degrees to 37 degrees. Changes in pH also can produce an increase in chromosome abnormalities. Changes in oxygen, so 21% versus 5%, 21% produces more chromosome abnormalities. Or changes in osmolality also have been linked to defects in meiotic spindle, which would cause aneuploidy. All these parameters, temperature, changing pH, oxygen, osmolarity, we know what's the optimal value, and this is only really if right now would be if the machine is malfunctioning or you never read any papers and you are just starting IVF. More on culture conditions. Fraguli and Warner, they studied if continuous versus sequential media, if it will have a difference in chromosome abnormalities, and both found that, for instance, the first found that mosaicism was higher when they were using continuous media compared to sequential media. And Werner did not find differences in aneuploidy, but they found more usable blastocysts when using continuous media. So this will suggest, these two studies, that continuous media is better at producing, implanting euploid embryos. And also, there's another paper on lactate, comparing two medias, one with high lactate and the other with low lactate. And the low lactate has a higher chance of producing euploid embryos. Meanwhile, the low lactate produces more euploid embryos, and the high lactate produces 10% less euploid embryos. And then there are contaminants. For instance, bisphenol A and bisphenol S are well known to produce aneuploidies, and these are two products found in plastics. So you have to aerate well these plastics. Make sure that the plastics that you use they don't have these components. Also, it has been suggested that how we do the biopsy can create artifacts that can perceive as chromosome abnormalities. So Kelk et al. Tried to see if an increased number of pulses of laser when we do the biopsy has an effect. They didn't find an effect, but Winnie et al. Found that poor biopsy techniques does result in more mosaic profiles. So that would be one reason in which a center not producing good biopsies could have higher rates of aneuploidy, although it's not real aneuploidy. It's just a bad biopsy. Also, there have been described differences in miscarriage rates between centers. This is data that from Sauerkar. And as you could see, there were significant differences from 2% to 23% miscarriage rate after PGT. Since all these embryos transferred were euploid, there has to be other reasons for that. And probably is how the progesterone was given in these centers. In conclusion, we have some evidence that control ovarian stimulation has an impact on chromosome abnormalities. It's in the live birth rates decrease with increasing gonadotropin dose, that euploidy rates vary among physicians due to the use of specific protocols, and that the fraction of euploid embryos increases with increasing fraction of HMG. But the absolute number of euploid embryos increases with the number of metaphase 2. Also, laboratory conditions such as culture media, pH, temperature, osmolarity do impact chromosome abnormalities, and then the different luteal support strategies may contribute to miscarriage. Beyond aneuploidy, now we're studying also metabolites. We can do metabolomics with very sophisticated machines that can detect up to 7,000 metabolites in picomole levels. And we have linked a series of these metabolites with implantation and with aneuploidy. As you know, even with PGT, only 70% of embryos implant, only 70% of euploid embryos implant. That's the highest that have been published. So there is still 30% of embryos that never implant, and we think that we can identify those with metabolites. We can identify them in the SPEN media of blastocysts cultured to day 5. And these metabolites, as you could see in these graphs, they are very black and white. Either they are present only in implanting embryos or absent completely in non-implanting ones, or vice versa. So here we have an accuracy of 97% regarding which ones will implant and will not implant. But also it could be done for aneuploidy, and we have metabolites that can detect up to 95% concordance with aneuploidy. This could be done for at least now for 2 different media. So there are several questions that we would like to ask with this new method, and is there an optical secretion of intake of nutrients? Does hormone stimulation has an effect on metabolomics? Can we help these studies to improve embryo culture media? and will be biomarkers of gender or metabolites that will tell us about gender and predict abortion. So we are still studying all this, but we think that this has a lot of potential to give us another aspects of why some embryos don't implant. And beyond human error, obviously, most of the things that we have discussed here are probably mistakes, human errors, or bad practice. And what obviously we want is that to increase the results in all the centers to the best centers, right? And for that, automation is going to be really valuable. You know that there is great variability between centers, from 20 to 60% aneuploidy rates in egg donors between centers, 10 to 80% pregnancy rate in egg donors depending on the center. Also methods and levels of skill between center to center produce these differences. And even the STAR and SART studies, which show no pregnancy improvement in PGT in patients younger than 35, despite we have 40% aneuploidy, means that there is a biopsy effect that probably it's also related to skill. So all these differences could be avoided if we had a fully automated IVF lab. And this is what we are developing right now. We're developing algorithms for control variant stimulation. We're developing metabolomics that also is fully automated. Oocyte vitrification and warming. This will be a machine that does warming and vitrification, a fully automated IVF lab, and a fully automated ICSI device. So with that, we hope that all these variations may decrease significantly in time. Thank you very much.